Welcome everybody. Sorry for this delay, few minutes. I'm Dr. Johani. I'm the director of the microbiology lab at the National Guard Hospital. And today, in this evening, it's a great event to talk about a very important topic. And I'm sure all of you facing a lot of issue in regard to sepsis diagnostics. So today with us, the team from T2 Diagnostics, talking about the T2 system and its role in sepsis diagnosis and antimicrobial resistance and antimicrobial stewardship program. I would like to start with Dr. Oja. She's the Chief Medical Director of T2 Biosystems. She's a graduate of medical school who's been working a lot with the lab quality management system and lab accreditation program, including ISO and CAB accreditation. Dr. Anjum, she's the regional integrated diagnostic solution at BD. And today she will be talking about the culture-independent testing impact on sepsis and antimicrobial resistance. Dr. Anjum.
Thank you. Thank you so much for the introductions, Dr. Samira, and thank you to the Saudi Society of Clinical Chemistry for the introduction and arranging this webinar. I hope I can be heard. You can hear me?
Yes, you can hear.
Thank you. And it's an honor to be participating as a speaker with Dr. Sahar and Dr. Samira Yu on this important topic. The topic has always been important. However, the relevance of sepsis and antimicrobial resistance has really increased with the pandemic. And we know that with COVID-19, there's been seen that there's increased rates of secondary infections and increased usage of antibiotics. I will turn the screen share on. Just give me a moment, please. Please let me know if you can. Please let me know if you can see the screen share. Is my presentation visible, please?
Yes, doctor.
Thank you so much. So I will be talking on culture-independent testing, which is very novel, and the impact. So as all of us know, sepsis is a burdensome condition across the world in terms of morbidity, mortality, and healthcare cost. Sepsis represents $62 billion in US healthcare cost, claims more lives each year than 3 leading causes of cancer deaths combined. CDC data states that each year, 1.7 million adults in the US develop sepsis. 270,000 Americans annually die as a result of sepsis. 1 in 3 patients who dies in hospital has sepsis. Another concerning factor is that hospitalized COVID patients, as compared to hospitalized influenza patients, face a 22% higher risk of developing sepsis and 113% more likely to experience septic shock. Antimicrobial resistance is on the rise globally and is one of the biggest public health challenges of our time. Recently, WHO identified it as one of the top 10 public health challenges of our times. CDC reports that each year in the US, more than 2.8 million people get an antibiotic-resistant infection, and more than 35,000 people die as a result. That means, on an average, someone in the US gets an antibiotic-resistant infection every 11 seconds, and every 15 minutes, someone dies as a result. Antimicrobial-resistant infections are estimated to cause 700,000 deaths annually worldwide, and recently, the data shows that the number is higher than this. It's estimated to be 900,000 deaths annually worldwide. The challenge of detecting sepsis-causing pathogen is that currently the gold standard is blood culture for detection, and it's an amazing technology and has been there for a long time, very reliable. However, the current gold standard is a race against time. As we all know, the time to result on an average is about 48 hours, and it can take between 1 to 5 days. Thus, there's reliance on empiric therapy, and which is optimal in 30 to 60% cases only. The impressive work of Kumar et al. States that each hour of delayed treatment increases the mortality risk by 7.6% during septic shock. If you look at this slide, this talks about why is blood culture actually requiring a 5-day incubation. So basically, if you could take just that example of Candida albicans, I'm not sure whether the pointer is showing you Candida albicans. Here you can say that the percentage recovery on day 2 is about 50%, and day 3 additional 30%, day 4 additional around 10%, and day 5 is again about 4 to 5%. So that means incubation is actually required for 5 days in order to not miss growth. The challenges of blood culture is that one is the timing, and the rest we'll discuss more. So the Surviving Sepsis Campaign R1 bundle requires obtaining a blood culture and starting antibiotics within 1 hour of patient presentation. Now, the dilemma is that blood culture positivity rates are low and the contamination rates are stated to be high. Also, administering antibiotics prior to specimen collection in blood culture and improper specimen collection like underfilled bottle, lack of site infection, suboptimal number of bottles can actually alter the results. So, because of all these challenges, it's seen that overdiagnosis and overprescribing of antibiotics is happening because overtreating is less harmful than missing a septic patient. Now, of course, all this comes at a high cost, high risk, because overprescribing, overtreatment leads to challenges like hospital-acquired infections, toxicity associated with antimicrobials, and continuing development of antimicrobial resistance associated with antimicrobial use. So this is really of significant concern. Please look at this slide. New CDC data shows that more than half of the antibiotic prescribing for selected events in the hospital is not consistent with recommended prescribing practices. And prescribing can be improved through optimizing antibiotic selection, using the shortest effective duration of therapy, and reassessing antibiotic treatment when diagnostic testing results are available. Thus, it's critical to understand that the sooner diagnostics test result is available, the sooner targeted therapy can be started, which will improve antibiotic usage and help fight antimicrobial resistance. There are multiple concerns with antimicrobial resistance. The number of people facing antibiotic resistance is high, too high currently, although it has been proven that prevention and infection control efforts can reduce deaths from antibiotic-resistant infection by 18% overall. As we already discussed, there's lack of early detection, because of which there's prolonged empiric therapy, and overprescribing leads to emergence and spread of antimicrobial resistance. Antibiotic stewardship plays an important role in optimizing the use of antibiotics. It's been shown by a CDC study that nationally more than 4.6 billion is being spent annually to treat infection caused by the multi-drug resistant germs. And if no action is taken, the death toll because of AMR could rise to as much as 10 million deaths annually by 2015 and cause a 3.8% reduction in annual GDP. COVID-19 impact on antimicrobial stewardship and antimicrobial overuse is very critical for us to understand. I would like to start by the statement from Dr. Ghebreyesus, the Director General of the World Health Organization. He stated that COVID-19 pandemic has led to an increased use of antibiotics, which ultimately will lead to higher bacterial resistance rates that will impact the burden of disease and deaths during the pandemic and beyond. It has been seen that the antibiotic prescribing in COVID-19 patients ranges from 58 to 100%. And the widespread use of antimicrobial therapy in cases of COVID patients is understandable, though it's very concerning, because 25 to 70% of severely ill COVID patients manifest evidence of sepsis. And it's very difficult to exclude bacterial or fungal superinfection just based on signs and symptoms, physical finding, radiology reports, when a timely, accurate detection is not readily available. The extended time from presentation to appropriate therapy continues to be a major contributor to poor patient outcomes and proliferation of antimicrobial resistance. And as we already discussed, that the current gold standard, the standard of care, is a race against times and can delay time-sensitive, life-saving targeted treatment. Now, the survival decreases by 7.6% during septic shock and for every hour delay in time to appropriate therapy. Also, prolonged use of broad-spectrum antimicrobials is a key risk factor associated with the development and spread of antimicrobial resistant organisms. We know that the rapid diagnosis and treatment could prevent as many as 80% of sepsis deaths. Now, looking at this slide, on the left extreme side is the time of sample collection, and it takes about 1 to 5 days to get a positive blood culture result. Now, the culture-based rapid diagnostic, multiple technologies, methodologies are available. However, these can provide sepsis identification in 1 to 3 hours only after the blood culture turns positive. Thus, they cannot be leveraged until the culture becomes positive and cannot be utilized in cases of culture-negative sepsis. As you know, culture-negative sepsis is a common clinical concern where the patient appears septic with a lack of pathogen growth on culture. Now, coming to non-culture-based rapid diagnostics, these are not reliant on culture positivity or influenced by antibiotic intake prior to the test. So these tests, specifically, I could talk about T2 Biosystem diagnostic panels. These are the only FDA cleared and CE mark test that's available and provides species identification directly from whole blood within 3 to 5 hours of the first blood draw with high sensitivity and specificity. And as we already discussed, that blood culture which is the current standard, as per the guidelines, at least 2 sets of blood culture bottles would be required, and every bottle ideally needs 10 ml or minimum of 8 ml blood in. So the time to positivity of blood culture varies and will take an average of 2 to 3 days, while T2 panel takes 2 to 5 hours depending on the panel used and requires only 4 ml of blood. The T2DX is the instrument that T2 Biosystems has, and you can see the picture here. So we need to draw 4 ml of patient blood in an 80-tier tube, load the blood tube onto the T2DX instrument, and the cell lysis, amplification of the target DNA, and the clustering of nanoparticles happens. This is a proprietary methodology with T2 Biosystems, which enables inhibition-free DNA amplification in a complex clinical matrices like the blood of a sepsis patient. It utilizes miniaturized magnetic resonance technology, which measures how water molecules react in presence of magnetic field. There's no background interference by human DNA, antibiotics, it simplifies the process and eliminates extraction and purification of targets. T2 magnetic resistance also enables measurement directly from patient sample, whole blood, enabling much higher sensitivity than other technologies. This is a very simple to use platform. It is FDA cleared. T2Dx instrument is FDA cleared. It's fully automated, walkaway, clinical multiplex, benchtop instrument. As you can see, there are 7 individual random access drawers. They can be loaded at any time. There's no batching required. There's a user-friendly touch screen, which displays, the display provides step-by-step guidance to the operator to load a sample. And as I already mentioned, there's no sample preparation required. There's minimum hands-on time per sample for the operator. The sample and the EDTA tube and the sample EDTA tube and the reagent tray snap onto the cartridge. You can load it onto the T2Dx instrument, and then it's ready to go, and it provides a species-specific result, which enables targeted therapy. The comprehensive product portfolio that's available for these sepsis patients is T2 Canada panel with the sensitivity of 91%, specificity of 99%, and it covers 5 fungal pathogens. These pathogens are known to cause more than 90% of invasive candidiasis. This is FDA cleared, CE marked, and the limit of detection is as low as 1 to 3 colony forming unit per mil. Going to the T2 bacteria panel, the sensitivity is 90%, specificity is 98%, and it covers most of the escape pathogens. Escape pathogens are a group of infectious bacteria that have garnered particular attention for their ability to escape or evade common therapies through antimicrobial resistance. These pathogens are resistant against common empiric therapies, and they are also known for causing majority of nosocomial infections. Also, a systematic review of clinical and economic impact of antibiotic resistance reveals that escape pathogens are associated with highest risk of mortality, thereby resulting in increased healthcare cost. There's a recent study which states that escape pathogens, when compared with non-escape pathogens, were associated with 3.3-day increase in length of stay, $5,500 increase in cost of care, and 2.1% absolute increase in mortality. So this is a very, very important panel for patients suffering with sepsis causing pathogen infection, bloodstream infection. This is FDA cleared, CE marked. The limit of detection is as low as 2 to 11 CFU per ml. And to mention, T2 bacteria panel is the first in vitro diagnostic test to ever receive approval from US Center for Medicare and Medicaid Services for new technology add-on payment. This helps an innovative platform to be used, leveraged more easily. Coming to the T2 resistance panel, please note this was granted breakthrough device designation by the FDA. The sensitivity and the specificity is more than 99%, and it covers 13 molecular markers of antibiotic resistance against the most common resistance that has been seen for gram-positive as well as gram-negative pathogens. And this is currently RUO, research use only in US, and we have already initiated the clinical study, multi-site clinical study across multiple centers in the US, and it is available OUS, out of the US, as CE mark, and the limit of detection is as low as 3 to 11 CFU per ml, which is very, very impressive. Now, this panel, the T2 SARS-CoV-2 panel, the PPA 95%, NPA 100%, this detects all the variants that have emerged till date. However, this is under emergency use authorization by FDA and available only in the US. And I'm just mentioning it for just covering all the panels that we have available at the moment. The COVID-19 secondary infection and sepsis, I just wanted to spend a little bit of time on this slide. So the secondary bacterial and fungal infections, including the antimicrobial resistant infections, are likely to occur in a clinically significant proportion of hospitalized patients with COVID-19, and it's been stated in multiple studies. And I didn't want to spend too much time on that, but I did want to mention one of the recent multi-center retrospective cohort analysis from 241 acute care hospitals of 590,960 patients evaluating COVID-19 and secondary infections. Some of the key findings was that SARS-CoV-2 positive patients were almost 2 times as likely to have a pathogen detected during the hospital onset period. The longer hospital stays and longer ICU stays for patients that tested positive for SARS-CoV-2 and a secondary pathogen. And you can see here, I already mentioned what are the pathogens that are available on the panel of T2 Biosystems sepsis panels. Here, the secondary pathogens which were identified most commonly have been covered, and the percentage distribution is also mentioned here. Staph aureus, E. coli, Candida albicans, Pseudomonas aeruginosa, Klebsiella pneumoniae, Candida glabrata. These are commonly seen, and these are available on the T2 panel. Now, there is growing real-world evidence demonstrating positive impact of T2 Candida and T2 bacteria on patient care and antimicrobial stewardship. Across hospitals, we note reduction in ICU and hospital length of stay, faster initiation of targeted therapy, as well as discontinuation of antimicrobials, and hospital savings. To mention a few, for T2 Candida, there's a study by Stuber et al., which includes about 628 patients in the US. Antifungal optimization was seen in 54% of patients, and antifungal therapy was avoided in 60% of negative cases. The T2 Candida negative patient had significantly fewer days of antifungal therapy compared to those with positive results. Patch et al. Did a retrospective analysis in the US, and the faster time to appropriate therapy was demonstrated, 34 hours versus 6 hours with blood culture. Unnecessary antifungals were avoided in 42.8% and discontinued after 1 dose in 15.6% of the negative patients. The average length of stay was reduced by 8 days, and average net antifungal saving was $280 for every patient tested. Now, for T2 bacteria, Seitz et al. Did a prospective study in Austria and demonstrated that T2 bacteria detected more pathogen, 41 versus 14% of the samples. There was faster time to species identification with difference of 59 hours, faster time to targeted antibiotic therapy, and shorter length of stay for T2 bacteria. Horowitz et al. They did a Can you hear me now? Can you hear me on the?
Yes, doctor. Yes.
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 Oh my God. I hope, which slides should I repeat? I will do it quickly. Should I repeat any?
18. 18.
18?
Yes.
Okay. So I'll go back to, please let me know you can hear me. So I'll go back to the slide sharing. Okay.
Okay.
Slide number 18.
Yes.
This is a slide? Okay.
No, the one before.
Before this.
Yes.
This one?
We are good. Thank you.
Yes. Thank you. Thank God I picked up the phone. Normally I don't. So I'm really sorry for I didn't realize what happened here. So there's growing real-world evidence demonstrating positive impact of T2 Candida and T2 bacteria on patient care and antimicrobial stewardship. Across hospitals, we noted reduction in ICU and hospital length of stay, faster initiation of targeted therapy, as well as discontinuation of antimicrobials and hospital savings. T2 Candida, I just wanted to mention two studies here. Stuber et al. 2020 did a retrospective study with pretty impressive patient size, subject enrollment size. Antifungal optimization occurred in 54% of patients with antifungal orders at time of T2 Candida test. Antifungal therapy was avoided in 60% of negative cases. Antifungal optimization. Can you hear me?
Yes.
T2 negative patients had significantly fewer days of antifungal therapy compared to those with positive results. Page et al. Did a retrospective analysis in the US, and faster time to appropriate therapy post T2 Candida was seen, 34 hours versus 6 hours with blood culture. Unnecessary antifungals were avoided in 42.8% and discontinued after one dose in 15.6% of T2 Candida negative patients. The average length of stay per patient reduced by 8 days. Average net antifungal savings of $280 was seen for every patient tested. Coming to T2 bacteria, Seitz et al. Did a prospective study in Austria and demonstrated that T2 bacteria detected more pathogens in the samples, 41 versus 14% of samples. There was faster time to species identification with a difference of about 59 hours, faster time to targeted antibiotic therapy, 6 hours with T2 bacteria versus 77 hours with blood culture, shorter length of stay for T2 bacteria. Horowitz et al. Did a prospective cohort analysis of hematopoietic stem cell transplant patients with febrile neutropenia in the US, and 29 patients out of 39 showed early de-escalation of empiric anti-pseudomonal therapy, and total of 124 days of anti-pseudomonal therapy was saved. Very, very impressive data. And there's been, this slide shows the recent clinical data on T2 bacteria and T2 resistance prospective studies. Please note that T2 resistance is not FDA cleared, as I already mentioned. It is used in the US only as research use. And right now, we already have the study, the clinical study for submission of FDA ongoing in the US, while it's available out of US as CE mark product. Paghi et al. In 2021 did a prospective study in Italy and showed that there was higher sensitivity and negative predictive value of 100% for the escape bloodstream infections for T2 bacteria compared to blood culture. And faster time to report 4 hours versus with blood culture 36 hours. Empiric therapy was switched the same day in 29% of cases, changed to directed therapy in 8% of patients with T2 bacteria negative result. Drevenik et al. 2021 did a prospective study in Czech Republic and demonstrated sensitivity of 94%, specificity of 100%, 55 hours faster than blood culture, and 9 of the 15 positive T2 bacteria patients had change in therapy based on the results. For T2 resistance, there's a prospective study of 57 patients in Italy and Greece. In Greece, there was faster mean time to positive result compared to blood culture with AST, 3.6 hours versus 101 hours, sensitivity and specificity of 91 and 90%, and changes in therapy were demonstrated in 22 events of discontinuation of unnecessary antibiotics and 10 events of escalation of antibiotics was shown. So all these studies demonstrate high sensitivity and specificity, faster time to result, escalation and de-escalation of antibiotics. And there are multiple other studies which are available. These were just few which we are sharing today. And I wanted to just quickly mention a very impressive publication, a meta-analysis of 14 controlled clinical studies on T2 magnetic resonance for rapid diagnosis of bloodstream infections. And this was published in Expert Review of Medical Devices. This is peer-reviewed, and the authors are from across the world. This includes about 7 US and 7 OUS studies, and this demonstrated faster time to detection and species identification with T2MR versus blood culture by 3 days, faster time to targeted antimicrobial therapy for patients testing positive on T2MR by 42 hours, and faster time to de-escalation from empirical therapy for patient testing negative on T2MR by 7 hours, shorter length of ICU stay and hospital stay by 5 days. And this is a systematic review of meta-analysis of control studies, multiple control studies. And I wanted to just quickly share with you one case of diagnosing candidemia with T2 Candida panel, an instructive case of septic shock in which blood cultures were negative. So this 27-year-old distant double lung transplant recipient who had cystic fibrosis developed fever, leukocytosis during hemodialysis with a new matured AVEF and discharged 5 days earlier. Patient has a previous history of tacrolimus-induced renal failure, a line-associated Enterococcus fecalis and coagulase negative staph, Candida glabrata, and Staph aureus bloodstream infection. The immunosuppression therapy consisted of cyclosporin, low-dose prednisolone, and vanco, and meropenem were given at hemodialysis and admission respectively. Now, T2 Candida was collected concurrently with blood culture 4 hours after microfungin dosing. T2 Candida positive in 4.5 hours for Candida albicans and Candida tropicalis. Antibiotics were discontinued. Patient eventually got better and was extubated within 72 hours. The ophthalmic examination on hospital day 5 and findings were consistent with Candida chorioretinitis, and microfungin was switched to fluconazole. Chorioretinitis confirmed the diagnosis of deep-seated infection due to hematogenous dissemination candidiasis. Treatment of candidemia is often delayed because blood culture, the gold standard diagnosis, is 50% sensitive for hematogenously disseminated candidiasis, and turnaround time is several days. So just to demonstrate that T2 Candida could be very, very effective because it can turn positive in 3 to 5 hours. In this case, it was 4.5 hours. And the right medicine could be started, right therapy could be started. And we know the challenge that in blood culture, it can be missed, as well it can take several days to detect. Another example for T2 Bacterial Panel, a 52-year-old female with polycystic liver and renal disease, history of several episodes of bloodstream infection due to Enterobacterialis, ESBL producing E. coli, Enterobacter cloacae complex, KPCK, pneumonia. T2 result, E. coli detected in approximately 5 hours, while blood culture remained negative after 5 days. The targeted therapy was initiated based on the T2 result, and the summary is that T2 magnetic resonance direct from blood detection of bacterial bloodstream infections showed 90% sensitivity, 98% specificity in clinical samples, and the ability for T2 Bacterial Panel to detect infections much faster than blood culture, very accurately. And this clinical study demonstrated ability of T2 bacteria to detect potential infection which could be missed by blood culture. And both these examples are real life cases, and the references are on both the slides. Now, till now we were discussing the panels which exist, and I wanted to share with you quickly the advancing pipeline that we have with multi-year government contract. We have new product development being enabled by about 70 million milestone-based funding by BARDA. BARDA is a Biomedical Advanced Research and Development Authority of the US government, Department of Health and Human Services Office. T2 resistance panel we've already discussed, and I won't spend much time on this, but this is also being supported by BARDA. Biothreat panel, this is a direct from blood test panel for detection of 6 biothreat pathogens, which are on the CDC A and B list for the biothreat agents. This can be detected from whole blood directly, and they're designed to run on FDA cleared T2Dx instrument, which is being used currently, and the clinical trials already been initiated, and we are doing very, very well and expecting very good enrollment. The 2 other panels which are under development right now are the comprehensive sepsis panel, which is direct from blood test panel designed to detect more than 95% of all bloodstream infections caused by bacteria and Candida, and antibiotic resistant markers identified as threats by CDC in a single test with a timed result of approximately 3 hours. This will be running on the next generation instrument, which is again designed to be fully automated random access like the current FDA cleared T2Dx instrument, designed in parallel with comprehensive sepsis panel to detect increased number of pathogens and the antibiotic resistant gene from a single whole blood sample. So the advantage would be you could use just one panel to do all the testing, and this would detect more than 95% of all the bloodstream infections. So I would like to end this discussion by sharing that together we can improve sepsis management and antimicrobial stewardship. We can get patients on the right therapy faster with tests which are like T2 Biosystem directly from whole blood, and currently this is the only test that we are aware of which can give you the species ID directly from whole blood, and it detects majority of the sepsis causing pathogens. Screens a suspected sepsis patient. 94% of healthcare professionals would test with blood culture first. That is a fact. It can improve outcomes. Potential reduction in mortality, morbidity have been demonstrated. Reduced length of stay, including ICU days, timely and targeted treatment, and less adverse events associated with the empiric therapy. It can also improve stewardship by reduced use of antibiotics, antifungals, rapid adjustment and de-escalation of therapy, decreasing the potential of antimicrobial resistance as the reliance on empiric therapy decreases. And also it helps in reducing the cost of sepsis management, cost reduction from early discharge, reduce ICU and hospital length of stay, reduce cost of antimicrobial, reimbursement of discharge ED patients. The things to remember are that rapid diagnostics is critical for timely initiation of targeted therapy. And the currently available culture-independent technologies are innovative. So the data shows the impact. So these should be leveraged, and blood culture remain the gold standard for diagnosing the bloodstream infection and are limited by slow turnaround time and sensitivity. The limitation of blood culture and the quality standard of sepsis lead to aggressive empiric broad spectrum antibiotic therapy, and this practice results in unnecessary antimicrobial usage, toxicity, and antimicrobial resistance. Culture-independent rapid diagnostic tests that detect pathogens directly from blood can transform clinical practice by decreasing resource utilization, improving clinical outcomes. It's very critical to understand for these innovative tests how to incorporate them into the clinical testing, into the lab workflow, into the clinical testing workflow, the treatment algorithms, because this is essential, critical for successful implementation. So the patient selection criteria needs to be very decided based on the hospital algorithms. And this is going to be very helpful when it is actually implemented into the lab workflow, into the clinical workflow and treatment algorithm correctly. And leveraging these tests on the patient on the risk of bloodstream infection and sepsis is going to have a major impact. So I'd like to end here by saying a big thank you for listening to me and providing me this opportunity. Thanks to all of you listening to this session. And please let me know if there are any questions now or later. Thank you. I'll stop sharing now.
Thank you so much. Thank you for this extensive review of the T2 system and the need for such diagnostic in a clinical microbiology lab. We will leave the question till the end of the session, and we will be moving to our next speaker, Dr. Sahar. Dr. Sahar is Al-Thawadi. She's the director of the microbiology lab at King Faisal Specialist Hospital and Research Center in Riyadh. She's a consultant microbiologist, and she's the microbiology lab director. And Dr. Sahar will be presenting King Faisal experience with the evaluation of T2 system in their lab. Dr. Sahar?
Thank you very much, Dr. Samira. Good evening, everybody. Can you hear me? Can you hear me, Dr. Samira?
I can hear very well.
Yes,
I can.
okay. Okay, thank you very much, Dr. Samira. I would like to thank initially the organizing committee for this invite. And I want to share just my presentation. Just give me a few seconds. Can you see my slides?
Yes.
Yes, okay. Okay, thank you very much. And what I'm going to be talking about in the coming, well, probably half an hour or less, just to share with you our experience with the evaluation of the T2 system. And I'm going to just talk about it, just to give you a very brief introduction. I think we heard a lot, extensive review of the T2 biosystem. And I'm going to tell you some of the limitation and the issues that we have faced during validation. We all know about the pitfalls of the current diagnostics, especially of sepsis. Diagnosis of sepsis is a very challenging entity. Blood culture remains the gold standard for diagnosis. However, it's with its low sensitivity and time consuming. And we all know that the targeted initial choice of antibiotic therapy really saves more lives than any other medical interventions. And this diagnosis should happen as early as possible, within 1 to 3 hours the faster the diagnosis, the better it is, from symptom-based sepsis recognition to the initiation of antimicrobial therapy, which we all know many studies showed that it can reduce the mortality rate. The emergence of resistant pathogens and the delayed diagnosis really lead to high morbidity and mortality. Now, what's the ideal diagnostic test that we have everybody I think wants to have? Rapid detection first in less than 3 hours, broad-based detection, including having bacteria, viruses, and fungi, minimal invasiveness, utilizing the clinical samples which you will usually collect for any septic patients with low specimen volumes, that you don't need higher sample volumes. You need a high sensitivity and specificity for the immediate initiation of targeted therapy. Polymicrobial detection of pathogens sometimes is important. Detection of drug resistance, ease of integration into any clinical workflow. It can be very simply to be operated. You don't need a complex system. And the ability to detect unknown and emerging pathogens. Now, some of these criteria is available in the current diagnostic testing, and we all have witnessed many changes in the field of diagnostic microbiology diagnostic lately. We are really moving away from traditional methods towards really a new generation of tests. It has a great potential to improve patient care. However, I have to mention that all the clinicians should be really mindful of innovation in their laboratory, including all the physicians. They need to know the sensitivity, specificity, advantages, and disadvantages of the test offered by their microbiology lab to inform appropriate test utilization and interpretation. They need to know the performance characteristics of the test that has been used in order to reflect this on their system. And there should be a really good communication between the lab and the end user of microbiology in order to better utilize those testing. Now, I think you've heard enough about the T2 system, and I don't want to spend much of more time, but I will just want to mention the difference between a T2 system is the first system that we have in the market that can use sample directly from blood sample that we are using that can detect sepsis directly from a blood sample rather than waiting for a culture like the other methods. So, and I think you've heard enough about its mechanism of detection. It's a cartridge-based system. It takes only 2 minutes to be operated, and you can see these are drawers, and you can test like 5 or 4 samples at the same time with different panels at each time. Now, we have heard also about the Candida panel, Bacterial Panel Resistant panel. You've heard about this. The Candida panel, actually, that was the first panel that has been utilized from this system early on, actually, with the immunocompromised or neutropenic patients. So this panel has really showed improved turnaround time. For the blood culture, takes 2 to 6 days, while the T2, within 3 to 4 hours, you can have a diagnosis. It really identified 5 Candida species: albicans, glabrata, Parapsilosis tropicalis, and Cruzia, which is the most common yeast pathogen that you can detect. It has a detection limit of 1 colony-forming unit per ml, which is really highly sensitive. And as you've heard in the previous lecture, that it has a really high sensitivity, 91, specificity, really higher specificity, actually 99%, and a higher sensitivity as well, with the detection sensitivities of 1 to 3 colony-forming units, depending on the Candida species. It has a really high negative predictive value, knowing the high specificity. The other panel is actually the Bacterial Panel, identifies 6 of the most common and deadly sepsis-causing species of bacteria, which is Acinetobacter baumannii, E. coli, Klebsiella, Pseudomonas, Enterococcus faecium, and Staph aureus, and which constitute really majority of a blood culture positive isolates. Now, for the Bacteria panel, it really detects almost 50% of all bacterial bloodstream infection. This is actually a clinical trial, has been conducted in 11 U.S. centers, multi-center study with 1,400 patients have been investigated, and the investigators actually have demonstrated that it has really highly accurate results, identifying 3 times more infections than the paired blood culture draw, knowing the mechanism of detection and the higher sensitivity of T2. It has a positive and negative result 2 days faster than the blood cultures. And that is actually where they found actually really a big opportunities to de-escalate therapy and improve the time to targeted therapy by days. Now, this is the third panel. We didn't have the luxury to test this panel yet. Hopefully, once it's released, it will be evaluated. This is again the first to detect bloodstream resistant markers, detect 13 resistant genes from both gram-positive and gram-negative pathogens directly from blood samples. And again, it's less than 10 colony-forming units detected for all targets. And it really, having the luxury to detect those resistant genes early in the blood really can prevent the spread and early isolation and targeted therapy for those patients harboring those resistant genes, which has really, it will reduce the mortality and hospitalization costs. Now, what did we do? Our evaluation at KFSH, at King Faisal Specialist Hospital in Riyadh, We did actually blood cultures from all ICU wards, adult ICU wards, were monitored for almost 6 months during the evaluation period. We have selected actually certain patients with the infectious diseases and ICU physician. So patients who met the following criteria were selected for T2 testing. Those patients with septic shock and immunocompromised status, like anybody presenting with septic shock plus immunocompromised status, like other, whatever you can define as immunocompromised, blood culture will be collected, as well as 4 ml of ADTA blood will be collected from these patients and sent to microbiology lab during 6-month period. So we had, during this month, we have 40 positive blood culture samples were collected and processed on T2 system, those patients that they had a positive blood cultures as well. Panels were selected according to grams. Like once we have a positive blood culture from these units, we would do gram stain initially from all positive blood cultures, and the panel of T2 will be selected accordingly. So if we see gram-positive bacteria, we're going to use the bacterial panel. If you see yeast, we're going to be using the Candida panel. And an extra 10 negative blood culture samples were included as a negative controls. So the following panels were done. We did almost 21 Candida panel, 19 bacterial panel, and all 50 samples were correlated with the blood culture results or the clinical presentation. Now, this is actually our data, and you can see here, these are actually the blood culture results, and you can see next to it is the T2 results, and here if it is compatible or not compatible. Now, in specimen number 5, most of them, they were compatible, they're detecting the same. In specimen number 5 and 6 and 7, the T2 actually has detected more than what has the blood culture detected. For example, blood culture for specimen number 5, we detected Candida krusei. However, T2 result, we detected Candida albicans, Tropicalis, krusei, and glabrata. However, the patient, when we went to the history, he had Candida albicans from his tracheal aspirate. Again, this is true for the other patients because we know the sensitivity of T2, so it may detected more than those. Some of those maybe did cells that has been detected. However, when we review the history, patient did had some of these positive yeast in other samples other than the blood. The same is true for isolate number 7. Now, we had out of these samples, we had only 1 non-compatible results, and which is specimen number 12. We had Candida parapsilosis from blood culture, while the T2 was not detected. Now, we could not explain this. Probably it's inhibited for some reason, or there was some problem in processing because at that time we didn't, like we freeze some of the sample, but for some times, or there was a delay in specimen delivery to the lab, which has been made. So something happened to this sample, was not detected in a T2. The rest of them, they were all compatible. And these are all the rest of the samples. So we did 21 sample, 20 were compatible results, and we had only 1 non-compatible with a positive predictive value of almost 95%. Now, when we moved to the bacteria sample validation as well, we did almost 19, and they were all compatible except 1. Again, we detected Enterococcus faecium and blood culture results. However, it was not detected in T2 results. So that was the only non-compatible results. Now, whatever the sample, I have to mention, whatever the sample volume that it's less than 4 ml, we actually added saline to make the sample up to 4 ml. This is one of the issues that we have faced. We got most of the time a little amount of sample. Instead of 4, we get 3, sometimes 1 ml or 2 ml. So we fill it up until with saline, and it was actually able to detect this. So that was an advantage actually for the system. So again, we did find similar to the Candida panel, like in this patient number 3, we did Klebsiella, we got Klebsiella pneumonia blood culture, but when we look at the T2 results, we got Klebsiella pneumonia with Enterococcus faecium. Now, the patient did have Enterococcus faecium in his peritoneal tissue. around the same previous time, actually before the sample on the 19th, for example, this one on 19th of March, and the T2 were positive on 27th of March, so previously. So probably he's still colonizing other type of sample, which was interesting. And the same is true for patient number 4, who had Acinetobacter in blood. However, he had an additional to Acinetobacter Pseudomonas aeruginosa, and he did had a history of both organisms isolated from blood run on previous samples. And the rest of the sample, they were all correlating. Blood culture results, was correlated very well with the T2 system. So we had a total of 19 samples. So the positive predictive value was almost 95%. Now, this is the overall performance. We did almost a total blood sample culture collected were 1,171. 1,131 were negative. We did 21 panel for bacteria and 19 only for for yeast. We did 21 and 19 for bacteria. And you have here the positive predictive value were almost 95% for both, 100% a negative predictive value with a sensitivity of almost 95% for both yeast and bacteria with 100% specificity. Now, what were the limitations of the evaluation? It was a very small number of samples evaluated during this time. There were actually difficulty in collecting the correct sample volume. As I mentioned, we always get like sometimes 1 or 2 or 3 ml of samples, and this actually was one of the limiting factors. This is why we have added actually saline to bring it up to 4. There was some delay in specimen delivery and processing, and we hope that this will be improved after we go live with the testing. And because of the limited number of targets detected by T2, this evaluation was guided actually by the positive blood culture gram stain. So we were waiting until the blood culture becomes positive, and then we were doing the T2. But hopefully on the real life, we will not be doing this, so we can see the value of T2 in detecting those pathogens. Now, the T2 biosystem will be implemented, hopefully. I mean, we did implement it, and we started actually last week of doing the test, and hopefully we're going to see the performance soon. We went live with the test almost last week. Hopefully, I mean, we did implement. It's going to be implemented next week at King Faisal, mainly in critical care units, transplants, and oncology wards. Can you hear me? Hello?
Yes, doctor.
Okay. So mainly, the test will be used mainly in critical care area, transplant, and oncology wards. The test will be ordered specifically will be limited only for infectious disease consultants and selected patients, like according to their clinical judgment. The test will be done immediately once it's received in the lab, as the automated bench will be operating 24/7, so we will not have any delay in the processing of these samples. Results will be verbally communicated and immediately reported in the system once it's having a positive or a negative, once it's positive. And the correlation with the blood culture will be monitored once the sample is reported by T2. Then correlation with the blood culture will be done once it is positive or negative. So in conclusion, diagnosis of sepsis really remains a challenging entity. Simple and easy technologies with minimal hands-on time that will allow easy integration into routine workflow and operation 24/7 will be highly useful. Implementation of these technologies will really lead to improvement in early diagnosis, targeted antibiotic therapy and isolation, which will lead to reduction in morbidity and mortality. And very important that appropriate utilization of these test methods is highly advised by developing actually a workflow in the lab. Immediate reporting of these results to the physician will be extremely important rather than batching them or doing them, because they're going to lose their flavor and their immediate benefits. So I'm going to stop in here, and I'm happy to receive any questions. And again, thank you very much.
Oh, can you hear me?
Yes.
Yes, okay. Thank you, Dr. Sahar. Thank you for the nice review of the system and your evaluation at King Faisal. And we are eager to hear the update when you go live on clinical surface. We are open for any questions or comments from the audience. So anybody, you can type the question if you like, or you can ask. Hello? If anybody has a question.
Can I add, this is Dr. Aparna Ahuja. Dr. Samira, can I make a
Yes.
comment just
Yes.
for, yeah. So thank you for this great presentation, Dr. Sahar. And the data is very impressive. And I also compliment you on the work that has been done. So since you said the T2 panels were implemented last week in your facility,
Yes.
have you seen any, because, you know, innovation, new test, is there any comment you have about your experience in the last 1 week? Is there excitement, or is there, like, you know, it could be a positive excitement to have new technology on board, or
Yes.
what
Yes,
are the
sure.
things? I
Yes.
would be.
Yes. Thank you for your question. Actually, we went live on Sunday or Monday last week, and we didn't receive any sample. We received one pediatric sample, and actually one of the physician was going to send it, but I was on leave. Actually, I'm on leave, so I'm going to go back next week to the hospital. I'm going to hopefully see the updates if there was any, because I'm still on leave actually for last week and this week. So hopefully by next week we can have some update on the usage, because we've made it very limited to the infectious disease physicians only. So hopefully on the next meeting, I'm going to have more stories to tell and more actual live cases with the T2 implementation. Thank you.
Thank you. And also, we'll be very interested to know the impact on patient outcomes.
Yes.
Would you be also able to describe you did mention, I noted that on your last slide, you mentioned about appropriate utilization. So, yes, a little bit about that diagnostic stewardship, where we do the right test on the right patient, and how that's why you're selecting specific patient population.
Yes.
So if you just explain that a little bit more for the audience, I think it'll help them.
I mean, unfortunately, I don't have any examples now, but if we're going to go live, I'm going to have more, because we decided actually with the infectious diseases that we're going to do this on selected units, on a selected group of patients, and limited for infectious disease. And we can see once this test is implemented, we're going to be monitoring this impact on the antimicrobial therapy, either de-escalation or escalation of therapy. We're going to be monitoring this actually once we go live with this test. And with the help of the Antimicrobial Stewardship group that we have in the hospital, because they are very excited about this test to be implemented. So hopefully we're going to have some utilization. What's the impact of this test on the escalation or de-escalation of antibiotic therapy? So we're going to be evaluating this within like 3 months data once we have it ready. And I think this will give us a power of the test and how did it contributed to this escalation or de-escalation. If that answers your question. Thank you.
Yes. Thank you so much. And I can say that we noticed that the impact of the test is higher when the right testing population is selected. So this is great information. Thanks for sharing.
You're welcome. Thank you.
Thank you. I don't see any question. Most of the questions about the Saudi Council. So I think we can conclude now. If Dr. Sahar or Dr. Anum, you want to add anything to the audience?
No, I'm fine. Unless they have any question, I'm fine actually.
And I would also say thank you. And I did provide my email. So if there are any questions in future, please, the audience can send it to me directly.
Yeah, sure. Thank you so much. Thank you for the great presentation.
Thank you. Thank you, Dr. Samira, and thank for the company, T2 organizers. Thank you very much. Thanks for the audience. Thank you.
Thank you. Thank you, Dr. Sahra, Dr. Samira, Maha, and the organizers, Saudi Society of Clinical Chemistry. Thank you so much.
Thank you.